There is a small crustacean living in shallow coastal oceans around the world that has evolved one of the most unusual parasitic strategies ever documented in biology. Its scientific name is Cymothoa exigua, and it belongs to a group of crustaceans called isopods, the same broad family that includes the pill bugs commonly found under rocks in gardens, though Cymothoa exigua has taken a considerably more specialized evolutionary path than its terrestrial relatives. What makes this organism remarkable is not simply that it parasitizes fish, which thousands of species do in various ways, but the specific mechanism it has developed and the organ it ultimately replaces. Adult females of this species live permanently inside the mouths of their fish hosts, attached to the site where the tongue was, functioning as a structural replacement for that organ. The fish continues to eat, move water across its gills, and carry out the mechanical functions that require a tongue, using the parasite as a substitute structure. This relationship has no close parallel anywhere else in the animal kingdom. The life cycle begins in open water. Juvenile Cymothoa exigua enter the world as males, small enough that they are vulnerable to being eaten by almost anything in their environment. Their immediate biological priority is finding a host before that happens. They detect fish through chemical gradients in the water, compounds released by fish skin and mucus that the juvenile isopod sensory systems are calibrated to follow. When a juvenile reaches a suitable host, typically a snapper or similar reef-associated fish species, it enters through the gill opening. The gills are the first residents, packed with blood vessels and providing a sheltered location with direct access to the circulatory system. Gill tissue represents a reliable feeding site for a small parasite that needs to grow before it can move to its permanent location. The juvenile attaches using hooked appendages and begins feeding on blood and gill mucus, growing steadily while remaining protected inside the body of the fish. This stage can last weeks to months depending on conditions, and during it the parasite is physiologically preparing for the transition that defines its adult life. When the isopod has grown sufficiently, it migrates from the gill chamber forward into the mouth cavity and locates the base of the tongue. Specifically, the hypoglossal artery, the primary blood supply that keeps tongue tissue alive. The parasite attaches its front legs to this location and begins drawing blood directly from the arterial supply. The consequence for the tongue unfolds gradually rather than suddenly. As the parasite diverts an increasing proportion of the blood supply, the tongue tissue receives less oxygen and fewer nutrients than it requires to maintain itself. Over time, the tissue atrophies. It shrinks progressively until very little of the original organ remains. In most biological contexts, the loss of an organ that performs essential mechanical functions would seriously compromise the animal's ability to survive. The tongue in fish is involved in prey manipulation, the movement of food toward the throat, and the management of water flow during feeding. Losing it without a replacement would be significantly damaging. What Cymothoa exigua has evolved is a solution to this problem that serves its own interests while incidentally preserving the host's functional capacity. Once the original tongue has atrophied, the isopod repositions itself, rotating and anchoring more firmly to the remaining tongue musculature at the base of the mouth, and effectively occupies the same spatial position and fulfills the same basic mechanical role. Research examining the feeding behavior of infected versus uninfected fish has found that many infected individuals feed with relatively normal efficiency, suggesting the replacement structure is functionally adequate for the fish's needs, even though it is a living organism with its own biology rather than the fish's own tissue. From the parasite's perspective, this outcome is essential. A dead host provides nothing. A functioning host that continues feeding provides a sustained supply of food. The parasite feeds on the fish's blood and mucus throughout its residence, and a protected environment in which the parasite is sheltered from predators and external conditions. The evolutionary pressure that shaped this strategy selected for a parasite that keeps its host alive and functional, which is a meaningfully different adaptive strategy from the more commonly recognized pattern of parasites that gradually weaken or kill their hosts. The reproductive biology of Cymothoa exigua adds another layer of complexity to an already unusual organism. Every individual begins life as a male. This is the juvenile stage that enters the host through the gills and eventually migrates to the mouth. If no female is already present in the host fish when the isopod reaches maturity, it undergoes a sex change and becomes female. A process called sequential hermaphroditism, or more specifically protandrous hermaphroditism, meaning the organism is male first and female second. This is not unique to Cymothoa exigua. Sequential hermaphroditism occurs in a number of fish and invertebrate species, including clownfish, which change in the opposite direction. But the context in which it operates here is particularly notable. The female, now permanently anchored to the tongue position inside the fish's mouth, represents the reproductive center of a small colony. If a second juvenile isopod enters the same host fish at a later point, it detects the chemical signals produced by the established female and remains male. The male takes up residence in the gill chamber rather than migrating to the mouth, and the two reproduce from their separate locations within the same host animal. The female broods the developing young internally, and when they are ready, the juveniles are released through the gill, opening into open water to begin the cycle again. From a genetic and evolutionary standpoint, the flexibility built into this system is significant. By starting all individuals as males, the sex that requires less biological investment to produce, the species ensures that every individual that successfully colonizes a host without an existing female can immediately fill the reproductive role the population needs. The sex change is triggered by social environment rather than by a predetermined genetic timeline, which is a form of developmental plasticity that allows the species to optimize its reproductive strategy based on the specific conditions each individual encounters. Studies on isopod populations in reef systems have found that infestation rates vary considerably by location and fish species, with some populations showing a surprisingly high proportion of infected individuals in areas where host fish are abundant, and juvenile isopod survival rates are favorable. It is easy to frame Cymothoa exigua purely as a biological curiosity, strange enough to be interesting, contained enough to seem distant from anything that directly matters. But the organism sits inside a broader ecological context that gives it more significance than its novelty alone would suggest. Parasites as a category are among the most numerically abundant and ecologically consequential organisms in marine ecosystems, and isopod parasites specifically are common enough in coastal reef systems that their effects on fish populations are measurable at the community level. Research published in marine ecology literature has documented that isopod infestation can affect the growth rates, reproductive output, and stress physiology of host fish populations, with implications that extend to the predators that feed on those fish and the prey species those fish consume. Cymothoa exigua in particular has been found across a wide geographic range: the Pacific and Atlantic coasts of the Americas, the Mediterranean, and Indo-Pacific reef systems, and across a broader range of host species than early descriptions suggested, including commercially important fish that appear in seafood markets. This is where the organism makes its most direct contact with human experience. People occasionally find Cymothoa exigua inside fish purchased at markets or caught recreationally, and the reaction is reliably strong. Part of what makes the discovery so striking is that it violates an expectation most people carry without examining. The assumption that the visible external appearance of an animal tells you most of what there is to know about its condition. A snapper that looks completely normal from the outside may have been carrying a living crustacean in its mouth for months or years. The fish is not diseased in the conventional sense, is not dangerous to eat, and shows no external signs of its unusual internal resident. What Cymothoa exigua ultimately illustrates, beyond its specific and genuinely extraordinary adaptations, is how much of the biology operating in the natural world is invisible at the scale humans normally observe it, and how the strategies that evolution produces under sustained pressure can be far more precise, far more specialized, and far stranger than intuition prepares us to expect.